Hi again. This video is going to be the first half of Unit 6, which was all about DNA, DNA replication, transcription, translation, and gene expression. So this first slide reviews for you the process of gene expression, which we call this the central dogma of biology because this process is common in all organisms. We go from our DNA, we transcribe that into RNA. RNA then gets translated into protein, and that protein is how our genes are expressed. Those proteins can be structural proteins, they can be enzymes, they can be all kinds of sort of different functionings for those proteins, but they are how our traits are expressed. I would just review the process. You would not really need to know the nitty-gritty details of transcription and translation, but just knowing that the enzyme RNA polymerase reads the DNA and creates a new RNA in the 5' to 3' direction. That's done by the enzyme. In eukaryotes, this is happening in the nucleus. In prokaryotes, this is happening in the cytoplasm because they don't have a nucleus. And in eukaryotes, before RNA leaves the nucleus, we have that extra step of RNA processing, where introns are removed and exons are spliced together. A 5' cap is added on that end, and a poly A tail is added on that end. And then this mRNA, the final product, will leave the nucleus, go out to a ribosome, and be translated. Now, if we're talking prokaryotes, a lot of times the RNA is translated immediately because there's nothing separating transcription from translation. But in eukaryotes, this has to leave the nucleus and go out to the ribosome. And with the help of tRNAs, our Ubers, this is how the amino acids are put together to build a polypeptide. And a polypeptide is, again, a protein. Again, I would be familiar with this chart or the wheel. You will never have to memorize this, but you may be asked to use it, and there is a question coming up where you will be asked to use it. Here is a list of some questions that you could possibly see on the AP exam. Again, we will go through quite a few of these today. And here is your slide with all of the different terms on it that we saw in Unit 6, or at least the first half of Unit 6. Now, let's again take a couple minutes to review some basic ideas about this. So we know that DNA and RNA are classified as nucleic acids. That's the group of organic compounds they are. And DNA and RNA are made up of nucleotides. A nucleotide is made up of a sugar, a phosphate, and a base. So here we have our phosphate group, we have our sugar. If it's DNA, that's going to be deoxyribose is the name of the sugar. And at this carbon right here, we would have a hydrogen. In RNA, the sugar is slightly different. It's called ribose, and the structural difference is that an OH group is there instead of the hydrogen. So we have our phosphate, we have our sugar, and then we have our bases. As you know, DNA has A, T, C, and G. A and G are the purines, which again, that just means that they're made up of two rings of nitrogen and carbon. And they also have C and T in DNA. They are classified as pyrimidines, and all that means is that they are bases with one ring. A purine always pairs with a pyrimidine. So C always pairs with G and A always pairs with T. That's going to give the shape of DNA and the structure of DNA. RNA has uracil in place of thymine. Again, that's another pyrimidine, and we know that when we're doing base pairing rules, A pairs with U instead of with T. Some other things to keep in mind, DNA is a double helix, so it's made of two strands of nucleotides, whereas RNA is only made up of one strand. Something else that we did in this unit was look at DNA replication, going from the 5' to 3' direction, where we talk about helicase, we talk about DNA polymerase, we talk about the single-stranded binding proteins, all of those different things that play the role in copying and replicating DNA. We know that at the replication fork, DNA polymerase can only work on the new strand in the 5' to 3' direction. So for this top strand, it's working this direction. On the bottom strand, it's working this direction. So the leading strand always is made towards the replication fork, and the lagging strand is made opposite. It's made going towards the origin, right? You kind of remember that, and we would see the same thing on the other side. So just a quick refresher of that. Now here's just, this isn't a AP question. This is just a question from one of the labs that we did together. We didn't really do anything super fancy for this lab. We just talked about it. But take a couple minutes, compare these different DNA strands, which species are going to be most closely related, which ones are going to be least closely related, and then looking at the different types of mutations. Okay, so if we're telling this evolution, we would know that organisms that have similar DNA strands or DNA codes would be more closely related. So comparing these, you should have seen that 2 and 3 were the most closely related because they had the most bases in common. Which species is least related? That's going to be 4 because it has the least amount of bases similar to the other ones. And then you could just pick any 2 strands and look at different mutations that you find. So following this along, these are the same up until this point right here, where we have an A, but we also have a T. That could have been a substitution, right? Because we still have all of the bases following that the same, things didn't shift one way or the other. So this is a substitution. We could even follow that further down and see if there's any other types of mutations. For example, if you look here, we would have another substitution, but the rest of the code seems to be pretty equal, okay? So that's just refreshing your memory on kind of tying that to evolution. Now, taking this to gene expression. We know that DNA is converted to RNA through the process of transcription. This is done by the enzyme RNA polymerase, and it's being made in the 5' to 3' direction. And we talked about this happening in different places in prokaryotes and eukaryotes. Here's a quick overview of what we talked about on one of those first slides with the RNA processing that happens in eukaryotes. So this is a nice slide to refresh your memory on that stuff we talked about a couple minutes ago. Here is a question from an AP exam way back in 2007. This one is just very straightforward. It's asking you A, B, and C. So take a couple seconds, read this, and then check your answer on the rubric on the next 2 slides because this one is pretty straightforward. Okay, so let's move into the more difficult one, which is going to be looking at sort of the second half of gene expression. We know that once an mRNA is made and it goes out to the ribosome and the cytoplasm, translation is the process that creates that protein from the codons. We know that we read the codons on the message. You always want to remember that. This chart comes from the messenger RNA. So we're using AUG from the messenger RNA. We would find that on there, and that tells us that that's start or methionine. Then we would move on to UUC. Using the chart, that's going to be phenylalanine, okay? It's the tRNAs that have the anticodons. Those are like the GPS in the Uber. That tells the tRNA where to bring the amino acid, right? So AUG is complementary to UAC. So this tells this tRNA to come to this spot. Same thing, UUC is complementary to this GPS, AAG, and that tells this tRNA to bring this amino acid. Okay, we talked about the different sites on the ribosome, the E, the P, and the A, and we even kind of did that silly little ribosome shuffle, where as a new tRNA comes in, everything shifts down, kicks this one out, and our polypeptide chain is growing on the amino acid that's held in the P site. We said A is for arrival, right? So this is that whole ribosome shuffle thing that we looked at when we talked about this process. Now, what would a question look like on the AP exam? Not to scare you, but this is what I was talking about on one of my prior videos, where they give you something that you don't know what it is, and they expect you to pick out, like, what are they really asking you? So take a couple minutes, read this, and then we will, so make sure you read both slides because there's A, B, and C, and then we will work through this problem together. So gibberellin, again, you probably don't know what that is, or GA3-beta hydroxylase. You don't need to know what that is because it tells you what it is, right? So this is the enzyme that's going to convert the pre-gibberellin to the active gibberellin. Gibberellin is going to be the hormone that causes plants to grow. Okay, so that's kind of, they give you the background information. We need gibberellin for a plant to grow. This is the enzyme that kind of creates that gibberellin. They then go on to tell you that there's a mutation in this gene, so in this enzyme, that results in a short plant phenotype. So that enzyme is no longer functioning. They then go on and talk about a Mendel experiment, so a monohybrid cross, and we see that by breeding a short plant with a tall plant, all the F1 are tall. Okay, that sounds a lot like Mendel, right? One of the traits disappeared, the other one covered it up. And then when we cross those offspring, there's a 3-to-1 ratio, with 75% of the plants being tall and 25% of the plants being short. Based on this information, it appears that the normal allele, this gene that's normal, is dominant, and the mutated allele is recessive, right? That's kind of what this sounds like. Now, part A, we're looking at the actual enzyme, and they're telling you that the DNA, the normal DNA, codes for a codon that codes for a non-polar amino acid, but the mutant allele codes for an amino acid which is threone, and they tell you that that's polar. I want you to think back to the party hat ridiculousness that I had you do. When we replace a non-polar, so that's going to be not water-loving, it's going to be on the inside of the enzyme, with a polar amino acid, that's going to be on the outside of the enzyme. What is that going to do to the shape of the enzyme? We know that if there's one little substitution of an amino acid like this, it's going to change the shape of the enzyme. Okay, so that's going to be your effect. Describe the effect. The enzyme will change shape and not function. And then the reasoning, how this mutation results in a short plant. Well, if the enzyme is not working, it's not going to be able to create gibberellin. And if there's no gibberellin, the plant will not grow, right? So it's like a chain reaction. So that's what part A was looking at. Part B, it's saying that if we are substituting alanine for threonine, what could have happened in the DNA? So we find our chart here, we find alanine is here. So any of these codes for alanine, and threonine, any of these code for threonine. What do you think happened? What's different between those two amino acids codons? If you look, it's the first letter, right? So if the first letter changed to this, any of those things there, that's probably what happened, okay? Part C, describe how individuals with 1 or 2 copies of the allele can have the same phenotype. So let's pretend that an individual is big G little g for that gibberellin enzyme. This is going to give a tall plant. We cannot tell if the plant is heterozygous or homozygous because both of these genotypes lead to the same phenotype. So they're asking you like how does that work? And what they really want you to think about is that as long as you're getting 1 copy of that good allele, you're going to have enough enzyme to make gibberellin. So just because it's, we have a mutated one right here, that doesn't mean anything. Just because we have 1 copy of it, it's still going to have the enzyme created to make the gibberellin. Okay, so the next couple slides, I think, walk you through that rubric, what you could look at, reflect on what I said, what you did in your writing, and there's the answers for that. Again, what I like at the end of all of these, they go through the checklist right from the AP curriculum and ask you some questions. So this is a nice place to reflect and say, yes, I know this, no, I don't know this. Tomorrow's video, you're going to watch the second half of Unit 6, which is all about gene control, and then you will practice those questions on AP Classroom. Thanks again.